and she is our licensed genetic counselor. Laura, are you available?
I am.
I will let you go ahead and present your slide.
Great. Okay, can everyone see that? Any issues?
Yes, perfect.
Okay, great. So as Nicole mentioned, my name is Laura Duncan. I'm one of the genetic counselors who works with individuals who have or who are at risk for various types of muscle conditions. And today I'd like to give an overview of genetic testing, kind of pull in some of the concepts that some of the other team members that presented today had brought up in their talks, and some considerations to take into account when pursuing genetic testing or after genetic test results come back. I do work at Vanderbilt University Medical Center up in Nashville, Tennessee, and do not have any conflicts of interest that are going to be impacting the information presented in today's talk. So some of the objectives for today is to go through basic genetics concepts, so get a little bit of genetics 101. So some of the terminology that was presented in previous talks might make a little sense in terms of common language, thinking about inheritance patterns, and then reviewing genetic testing options, as well as the importance of pre-test counseling, meaning understanding what you're being tested for and why, and what the potential outcomes are of that test. And then finally discussing the role of a genetic counselor in this process and ways to find genetic counselors if that's going to be part of your diagnostic journey or a tool that you need post-diagnosis. So to get started with some genetics 101, some of you might have seen images similar to this one, but this image just gives a little overview of some of the generalized concepts of genetics. So you might remember that in each of our cells, we have a set of chromosomes. If we take this chromosome and unwind it, that's just a long strand of DNA. And so we essentially think of our chromosomes as the packaging of this DNA that tightly coil it around so that it's able to fit in each cell. When we think about our DNA, when we're referring to a gene, we refer to a specific section of DNA that the body literally reads, much like you would read a recipe out of a recipe book, to make the protein product. And that protein product is what goes off and does the job in the body. And as you heard from some of the talks earlier today, there are various genes that can impact various proteins that are important within the muscle. So there are proteins that help with muscle communication, proteins that help with muscle stability, muscle integrity, and they might be linked to various genes that can lead to the same clinical story. So potential limb-girdle muscular dystrophy, Emery-Difus muscular dystrophy, even myotonic dystrophies, those kind of clinical diagnoses, but they might have a different genetic mechanism behind them or a cause to that specific muscle condition in that individual. And so if we take an even closer look at the genes, you'll see that we typically have these little building blocks of DNA, essentially the rungs across this DNA ladder that we're seeing in this picture. And we just abbreviate those building blocks by their initials. A, T, G, and C are the initials for each of these building blocks. You might have heard of them as nucleotides or base pairs if you're thinking back to science courses that you might have taken in the past. But essentially, they're the alphabet that tells the body what pieces of the protein to add next. So the body will identify the beginning of a gene by reading along the DNA. There'll be a signal to start the reading. It will start reading that DNA and be using the letters in groups of 3 to build each individual building block of that protein product. Proteins can be various sizes, and they all have different instructions to tell them how to be made appropriately. Everybody has changes in their DNA. So everybody in this world that we were to test would have many, many changes from each other. That's why we're not the same. We're each unique. But sometimes those changes impact the body's ability to read the DNA, cause that protein to be made incorrectly, and can lead to a genetic condition such as some of these muscle disorders that we've talked about so far. We've mentioned exons, as well as might have been mentioned introns as part of this conversation. So I thought I'd just show this little picture to kind of depict what an exon is. So it makes a little bit more sense in context of what we've talked about so far. So once more, this little image here is going to depict that DNA. Pretend these little kind of boxes along that kind of ladder that we're building are those base pairs or those building blocks of DNA, that alphabet. You can see here that the body actually cuts out portions of the DNA as it reads along because it doesn't need the full strand of DNA to make that protein product. So as the body finds its start, it reads along a portion of the DNA called the exon. There will then be a signal to the body that says, oh, the exon has ended. We don't need this portion of DNA next, so this blue section here, and we're going to cut and link to the next exon signal. And it will then read through that exon, hit a signal to say, oh, we don't need this section of blue DNA, and cut across to the next exon signal. This process is called splicing. It's essentially cutting the gene and putting it back together again, so it's a much shorter read that we would look at to actually be read to make that final protein product. So when we think about the exons within our DNA, we're essentially talking about the parts of the gene that make the protein, the actual reading instructions, the manual to tell you what to add next. And the introns are what we refer to as those kind of fillers in between the exon. The vast majority of our DNA is not used to make protein product. Only 1 to 2% of our DNA is used to make protein product. So earlier it was asked about exome sequencing. Essentially, that's reading the exons of the DNA, but could potentially miss changes that are at the intron level. So that kind of goes through a few of the definitions related to our genes. Another important concept related to genes is that you typically get 2 copies of each gene. Half of your genetic material that comes from your biological mom and half of your genetic material that comes from your biological dad. And so in each kind of cell, we get a set of chromosomes from mom and a set of chromosomes from dad. And when an individual goes on to make the sperm or the egg that would make their future children, they would only pass on half of their genetic material. And so these pictures here just depict an individual cell for an individual who is assigned female at birth, and a cell for an individual who's assigned male at birth. Typically, individuals assigned female at birth, so with female genitalia and or female characteristics, will have 2 X chromosomes. Those are what we call our sex chromosomes. They have 2 copies of the genes on this chromosome, while males assigned at birth will have 1 X and 1 Y. As you can see, the X and the Y are different from each other. Therefore, males only have 1 copy of the genes on the X chromosome. For the rest of our chromosomes, and therefore the genes on the rest of these chromosomes, both males and females will have 2 copies of those genes, typically. Okay, so as I mentioned, when thinking about the DNA, we can have genetic changes, or what we refer to as variants, or mutations that occur at the DNA level. And these changes might be classified differently. As I mentioned, we all have thousands and thousands of genetic changes, most of which fall in this category called benign. This essentially means that that change is not impacting the protein's ability to do its job. The body can still read the gene in the way that we would expect for it to. The protein is still made correctly, and it can still go off and function in the body. These also tend to be very common changes. So there might be genetic changes that influence the uniqueness between you and I, but they won't be leading to a genetic diagnosis necessarily. And the vast majority of genetic changes that occur are these benign changes. When we find a genetic change, we could also classify it as pathogenic. This word means that this genetic change can impact that protein product. It can potentially be a risk factor for a genetic condition, and it tends to be a rare change in the population. There are some genetic changes linked to disease that are a little bit more common, but we can use a lot of different pieces of information to help us designate which genetic changes are harmless and which genetic changes might be a risk factor for a genetic disorder. And then, as one of our speakers earlier mentioned, we also have this lovely gray zone of uncertainty that falls in between. This is called a variant of uncertain or unknown significance, meaning I see a genetic change. I know it's different from what we typically see in that gene, but I don't have enough data today to know if it could be associated with the symptoms for that individual or be a risk factor for that individual, or if it's a completely harmless change. And the more testing that we do, the more likely we are to find a variant of uncertain significance, but also eventually we're hoping to be able to classify it better into one of these more defined categories of risk factor or not. Okay. And then finally, when thinking about genetics, we know that genetic conditions can run in families in different ways. So there are genetic conditions that we call autosomal dominant, meaning it's on the chromosomes or in the genetic material that both males and females share equally. And these conditions require only one pathogenic or disease-causing change for an individual to be at risk. And so since I mentioned at the beginning, we have 2 copies of each gene. If you have a genetic change on one of the 2 copies and were to go on and have a biological child, you would then have a 50% chance to pass that change on to your child, and they could have the same genetic condition you do, or a 50% chance to pass on the non-changed gene, aka the working gene, and that individual would not be at risk for the same genetic condition you have. And so when we think about dominant inheritance, it's that 50-50 chance. Boys and girls can be impacted equally because it's on the chromosomes that both boys and girls share, and it only took the one change to lead to the disorder. Controversial, or I guess on the opposite spectrum of that, we also have genetic conditions that we call recessive. These are conditions that require 2 pathogenic changes to be at risk for the condition, 1 on each copy. So you can actually have a genetic change in this gene on one of your 2 copies, but the other one is a working copy of that gene, and therefore protects you from having the condition. You might be what we call a carrier for the condition. But if you were to pass on your gene change and a partner were to pass on his or her gene change, and your child got not 2 non-working copies, that child could be at risk for the condition. And so some genetic conditions you can actually be a carrier for, not show signs or symptoms, but have a chance to have a child with that same condition. And finally, some genetic conditions are what we call X-linked. This means that they're located on that X chromosome that I mentioned, that sex chromosome, that typically females will have 2 copies of and males will have 1 copy of, which might mean that an individual who's assigned female at birth, who has a genetic change in this gene, could be considered a carrier for it because she has that backup working copy, while an individual who is assigned male at birth, who only had the 1 X, if he has a change in that gene, he might actually show symptoms of the disorder since he does not have a backup. And it's not important necessarily to know all of these different inheritance patterns or all these different genes and types of changes, but it's important to know that when genetic testing occurs, it might have different impacts for you in terms of future family planning, or for your other relatives in terms of what are their chances of having the same thing, and what would be the utility or the need for genetic testing for those individuals. We typically know which genes go along with which inheritance patterns, and there can be clinical diagnoses, like the limb-girdle muscular dystrophies, where you can have different subtypes that lead to different, or that are linked to different inheritance patterns. So that's where genetics can be really useful in terms of thinking about impact for the family. Oops. Finally, a basic genetic concept that I wanted to make sure y'all are aware of is we actually have a second set of genetic material that's within each of our cells. So we've been talking about the DNA that's in that nucleus of the cell, packaged tightly in those chromosomes. That's where the vast majority of genetic conditions are linked to, what we call our nuclear DNA. But we can also have changes in the second set of DNA in each of our cells called our mitochondrial DNA. Y'all might remember Your flashback high school biology might have tried to block that out of your mind, but it's coming back to you. Mitochondria is the powerhouse of the cell. It's where we create a lot of the energy that the body needs. And it is what has its own set of DNA. And so the genetic transmission or the inheritance of mitochondrial DNA is a little bit different than what we talked about for the nuclear DNA, but important to keep in mind because mitochondrial conditions can have muscle clinical symptoms. And so sometimes if we focus the DNA and the nuclear level and that testing is normal, based off of your or your family's signs or symptoms, we might want to also assess the mitochondrial DNA, which tends to be transmitted from biological mothers or egg donors to the child, as opposed to being a combination of biological mom and biological dad or egg and sperm put together. So we go over all of this and you're like, wow, that's a lot. Basic Genetics 101 is a lot of little pieces of information. And the reason that I mentioned all of these different types of genetics and all of these different inheritance patterns is that muscle conditions vary in terms of the genetic background. They can be dominant, recessive, X-linked, mitochondrial. They can be caused by various types of changes at the DNA level. So we've talked a little bit about deletions within the gene, so having a missing piece of genetic material. Muscle conditions can be caused by duplications within the gene or an extra piece of genetic material. They can be caused by a misspelling or a sequence change. They can be caused by repeats within the DNA, much like myotonic dystrophy, which is brought up earlier today, where the same several letters of the gene are repeated over and over again. And in some individuals, those repeats get too large and the body can no longer read and produce that protein product the way that it can do its job. And so there's a lot of different kind of backgrounds of various muscle conditions. Not all muscle conditions have a single identifiable genetic cause. So even if we see something tracking in the family, we might do all the genetic testing available and not be able to discover the cause for that family because our knowledge has not caught up to our technology. We have about 25,000 different genes in our body that we know of to date. And we have only linked about 6,000 of those genes to an actual disorder, which leads to 20,000 genes where we still don't know what they do and if they might be linked to a genetic condition in humans. Every day, though, we discover new linkage, new associations, new understanding of different variants that pop up. So this technology and our knowledge is always improving, which is why it's important, even if you had genetic testing 10 years ago or 5 years ago or even 2 years ago, that was normal, to continue to have a relationship with a provider who's familiar with genetic testing because we might have new advances that could find a diagnosis now. And also a big important point here is even in one family, we can see variability in the presentation of these conditions. So you are more than just one gene. So even if you have a genetic variant or genetic mutation that you share with your mom or you share with your child or you share with your brother or your sister, y'all are all going to be unique from each other. And we can see different timelines for individual, different symptoms, different kind of courses of disease. And unfortunately, genetic testing doesn't always help us predict exactly the future for any individual because we are all so unique. Okay, so we talked about Genetics 101, gone a little bit about inheritance, talked a little bit about types of genetic changes. Let's talk more about genetic testing options. All right, so there are many different types of genetic tests, as you might have experienced personally or kind of picked up in hearing our conversation today. Depending on your clinical symptoms, depending on your family structure, if there's anyone else who might have the same symptoms, if we see a pattern in the family that can help us align better with a specific diagnosis or not, your genetic testing might be different than your friend's genetic testing, even if he or she also has a muscle condition, because once more, we are all unique individuals. If we find a diagnosis in a family member, and you want to know your chance to have the same muscle condition or to be a carrier for it, we can do very targeted genetic testing based on that family member's test. So it's very kind of tailored into the specific variant in that family. We also can test for individual genes at a time. So for example, myotonic dystrophy is due to the gene DMPK, or at least type 1 is, and we can test DMPK by itself. Or if we have suspicion that an individual has Duchenne's or Becker's muscular dystrophy, we can test DMD, the gene linked to that muscular dystrophy by itself. Or we can do something more extensive called a panel. A panel is when we take groups of genes and link them together, because it's hard for us to clinically know based off of the symptoms or even based off the family structure which gene we want to target. And so panels are not all created equal. You can have a panel called the limb-girdle muscular dystrophy panel or the Emery-Dreifuss muscular dystrophy panel that has 2 genes on it. You can have a limb-girdle muscular dystrophy panel that has 20 genes on it. And so it's important to know and to discuss with your provider exactly what's being tested, because it's possible your diagnosis is because of a gene not on that targeted panel. It was also asked earlier about exome sequencing. So remember we talked about the exons being the protein coding parts of the DNA? This, instead of picking and choosing different genes to look at, is kind of like a screen of those 25,000 genes to see if there's any genetic changes that are obvious. I agree with, I think it was Dr. Kahn who said that exome should not be our first tier test, because what these panels will do is actually make sure they cover our genes of interest in a lot of detail. So they might have a better coverage of the genes that we care most about. Exome is more of a screening test that looks at all of the genes but might miss portions of the DNA in the genes that we're most interested in. So I would also recommend we start out with a targeted test, as targeted as possible based off of your clinical story or your family structure, and then consider exome for some individuals down the road. Exome sequencing isn't perfect by any means, so it's very important to talk to your provider about the best test for you. We also want to make sure that when we're looking at genetic testing options, that if we're suspicious of certain types of genetic muscle conditions, that we are getting a look at the material to see if there's missing or extra pieces. So sequencing means we read the DNA and look for misspellings, while deletion duplication means we're looking for missing or extra pieces in the DNA. And so you also want to have an understanding of what were you tested for? Sequencing changes, deletion duplication changes, a combination of the two, because you could have negative deletion duplication studies and have a sequencing change, or have negative sequencing studies and have a deletion or a duplication. So really important to know the technology of the genetic testing. We also might do biochemical tests. So this isn't looking at the DNA itself, but looking at the products that that protein might be breaking down or the muscle might be breaking down to give us clues of what to target. And so this could be a CK level, this could be an acylcarnitine profile, but these can sometimes be linked to various genetic conditions and be a helpful clue to help us decide what to target in our testing. All right, so there's a lot of testing options out there. There are hundreds of labs that offer genetic testing, and each lab offers hundreds of genetic tests. So there are many, many choices, and no two choices are the same. So test selection is really important to understand when it comes to working with your provider. What are you being tested for? What could it miss? What does this mean for you? And potentially what are their costs, because they might all vary. Before the test is ordered, you should have a conversation with your provider about the types of results that can come back. Positive, negative, these variants of uncertain significance, and have a good understanding of that. You should understand if it can find things outside of the muscle diagnosis that you or your family member might have. You should also have an understanding of how these results could impact the family. So genetic testing can be a wealth of information to know the why, the what does this mean for my future potentially, but can also have some pretty significant emotional impacts because of the impact on family, knowing it might be passed to your children, or knowing that it came from your mom or your dad, or just the kind of emotions of having a diagnosis that might lead to more complications than you currently have. So keeping into account that this can be family news and can have an impact. There are benefits for genetic testing. So we've touched on several of these earlier today, including research eligibility, Does your gene of interest and the specific type of variant in your gene have treatments that are coming down the pipeline? Does it have treatments that are here now? Are there other risk factors for you down the road in terms of thinking about cardiac or hearing or kidney? And then kind of answering the unanswered question of why for a lot of individuals. And then also being able to find that community support. I think that MDA is amazing, and it's very general in terms of the support that it provides for individuals with various types of muscle conditions, and does have very targeted support as well. And then there are other community resources that might be more tailored to a specific diagnosis, where you can talk to individuals who have the same thing that you have. But genetic testing also has its limitations. You need to know what can be missed with this test, and understand that the testing will not always change your day-to-day. Your diet needs, your PT, OT, speech, all those day-to-day things that you might be doing might not be impacted by your genetic results, because those are based off of you and who you are and what symptoms you might be showing. And although genetic testing might give us some information compared to other people who have the same diagnosis, you are uniquely you, and we can't put all of our eggs into the genetic testing basket necessarily. So keep in mind that it might not change what's happening for you on a day-to-day basis, nor does it always lead to research or treatment. So kind of managing those expectations related to the genetic results. You also want to make sure you know how long results are supposed to take. So depending on the test, it can take anywhere from a few weeks to a few months. It's not a test that we get back the same day. So ask how long it's supposed to take, and then follow up with your provider if you haven't heard back within that time frame. There can be delays to testing, so it's helpful to know if it's taking about 4 weeks, check in about 4 weeks so they can check in with the lab and see what's going on. And then thinking about the cost and coverage. So when it comes to cost of genetic testing, depends on the test, the company you're ordering to, your insurance plan. So as mentioned earlier, these tests are more and more likely to be covered by insurance, but not always. And even if they are covered by insurance, you still have to pay your deductible, any co-pays, all the way up to your out-of-pocket max before the testing might have a lower cost or no cost. It also might depend on the hospital that the testing is going through or the doctor that's ordering the test. Sometimes they have to send through a hospital that has hospital processing fees, so it's important to know about that. Sometimes they send directly to a laboratory that does the test. It's important to know about insurance or self-pay options. So having a conversation about the finances behind genetic testing is very important. It was mentioned earlier about some free testing programs. Remember that free might have caveats that you need to be aware of. Most of the time, this means your information is being shared with others. So knowing who gets your information and what type of information they receive is a really important pre-test counseling aspect, because you don't want your information traveling to places that you don't know it's going to. And so when you are using sponsored tests by these big companies that are offering offering treatments, you got to know exactly what's happening to your information. Because some individuals are worried about their risk for discrimination. So we do have a law called GINA that protects you from your health insurance company and your employer from either dropping you or firing you based off of your genetic data, but that does not protect you from discrimination by long-term disability, short-term disability, life insurance, some of those specialty insurances that individuals who might have a muscle diagnosis might need down the road. All right, this is complicated. So where do you go to talk to someone about your specific story or your specific diagnosis or your specific concerns? Well, you can ask for a local genetic counselor. So there are about 5,000 genetic counselors throughout the country, and that number is increasing because the number of genetic tests that are getting ordered are increasing. We're trained in the medical aspects of genetics as well as the emotional and psychosocial, so the counseling aspect. And our job is to guide and support patients seeking information about the condition, about their family, about genetic tests, about what this means for their future, about resources. We can be located within an MDA clinic or outside of an MDA clinic as part of a hospital or clinical team. And we can also be located in other aspects of your care, in an insurance company, in a laboratory, in the public health sector, thinking about the newborn screening, things like spinal muscular atrophy. So genetic counselors are all over the place. but clinically, it might be helpful to talk to a genetic counselor about your concerns. How do you find a counselor by you? Well, we have this great website called findageneticcounselor.com. So you can go to findageneticcounselor.com and be linked to a genetic counselor locally, or talk to your local neurologist or your local genetics team if you have one already, or your local MDA clinic to be linked to someone who could kind of walk you through some of this, both the pre-test aspect and then potentially the post-diagnosis. Our job is to specialize in rare. So you might have a rare diagnosis, and you're like, well, no one knows about it. You're right, I might never have seen a person with it, but my job is to learn all about it so I can become a mini expert on that disorder, talk to you about it, and be able to summarize what we know to talk to you about it, even if I've never heard of it before, or if I've never talked to another person with that same diagnosis. We also have genetic counselors who specialize in muscle world. So you could find one of them and they might have multiple individuals with the same diagnosis that they could also link you with. When you see a genetic counselor, they'll ask you a lot of questions. Your personal history, your family history, about your emotional journey, about your diagnostic journey, but they'll also give you a lot of information. Testing options, helping select a test, talking about test results, talking about limitations of testing or outcomes of testing on the back end. And so I went through and also grabbed a couple of resources that might be helpful for individuals who are thinking about genetic testing or have already had genetic testing and have questions, or might have questions such as a question earlier about the impact of genetic testing on them. Even though it was found 20, 30 years ago, we can still help address some of those things. So some of these resources about genetic counseling tells you a little bit more about genetic counselors. Find a genetic counselor, that's how you find one of us. Genetics Home Reference is one of my favorite resources. It is breaking down genetics concepts about disorders, about the genetics behind it, about the inheritance behind it, in a way that's so easy to understand. I go there whenever I'm learning something new. They're moving over to Medline on October 1st, so I don't know what it's going to look like, but I think the information is supposed to be the same. Gene Reviews is written by geneticists for geneticists, so it might be a little TMI, but it can be a useful tool when thinking about management and give additional information about the clinical story. Finding condition-specific support groups or resources. We mentioned earlier clinicaltrials.gov can help find research opportunities. And then the MDA has lots of information about genetic testing and some of these genetic diagnoses linked to muscle clinical symptoms. And so there's a lot of resources out there for you. Make sure that if you have questions, you're having these conversations with your providers and getting hooked up with the appropriate team. And with that, if there's any additional questions, I would be happy to answer them.
Thank you, Laura. We did have someone submit a question. He says he's a male and has FSHD, probably a mild case. My adult son shows no symptoms and he is 40 with 2 small children. Could he have carried the mutation and passed it to them with the possibility that they will manifest symptoms when they are older?
That is a great question. So you bring up a great point. Some muscle diagnoses do not show signs or symptoms until much later in life. And so sometimes individuals can have the gene change, but not show symptoms and may show symptoms down the road. It can be really important based off of the genetics of your FSHD and kind of your diagnostic journey, your test results if you've had genetic testing, to offer your son to talk to a genetic counselor about his personal risk, which could then give information about your grandkids. Because of this later onset nature of some conditions, we might not know someone has the gene change until much later in their life.
Okay. And if someone would have, such as limb-girdle 2A, can their child have a different subset?
So typically, if the gene will stay the same as it passes through the family, so the subset should in theory stay the same, but the clinical symptoms or the clinical story of that individual might differ because other genes might be interacting with that gene, and I don't know limb-girdle 2A off the top of my head which gene that is, but other genes might interact with it. Your environment, the type of support you got through PT, OT, diet, etc., can all influence the story. So the subtype should stay the same, but the story might be a little different.
Okay. I'm just going through the chat here. I'm not seeing any other questions, so I appreciate your time today.
Yes, thank you. And
thank you very much.
Use some of these resources.
Yes, that's great. That's a great resource. Thank you very much.
Thank you.
And this would conclude our seminar for today. I want to thank all of our presenters for their time and effort they spent in putting together all their presentations. I also want to thank again our sponsors, Biogen, New Motion Foundation, and Genentech. And thank you to all you for submitting all your questions. We had a ton of questions today, so it really makes the symposium so much more beneficial for everyone. And if you do have any questions after this, please let me know. You can email mdaengage@mda.org, mdausa.org, sorry. And then finally, I want to remind everyone that I will be sending a brief survey to everyone who was viewing today. So I appreciate you taking 5 minutes to send us back your feedback and let us know what you thought of today's program. And thank you again, Dr. Sanka, for the keynote presentation. And that is it for today. So you guys have a great rest of your weekend.